Today we will continue our subject on the acquired immunity. So we talked about the importance of antibodies released from the activated B lymphocytes. They can help our body to protect us from the invading pathogens by several mechanisms. The first mechanism is by agglutination, when the antibody binds to the antigen in different places or binding sites, and each antigen is bound to several antibodies, then this reaction will cause clumping of the pathogens or the antigens, okay, and that's what we call agglutination. Also, this leads to precipitation of the and isolation of the pathogen or the antigens that cause the inflammatory reaction away from being distributed in the tissues or outside the tissues. Also, the immunoglobulin binding on the pathogen, they cover all the, let's say, the surfaces of the pathogen, leading to neutralization of the microorganism or the pathogen. And this actually helps to reduce the harmful effects from the microorganism. And also, immunoglobulins can cause lysis of the cells, damage and destruction of the cells, as well as the antibody antigen reaction will activate a complement system, which we will explain now. The complement system activation aggravates and help the immune cells as well as the antibodies to increase their efficiency in protecting us in inflammatory response. So, okay. Okay, so this is how agglutination occurs, as you can see here. These are the microorganisms or the antigens, and binding of the antibodies to these will cause clumping because of the many binding sites that each antibody exhibits. Of course, when the antibody is if the antibodies have more binding sites, it's even more clumping. Okay. The complement system is actually a system or several components which are actually proteins that can act like enzymes, are present in our plasma as inactive enzymes or proteins. Binding of the antigen to the antibody, okay, will activate a cascade of reactions leading to activation of components of this system. The complement system called complement because it's complementary to the role of antibodies and the immune cells. Okay, it helps their functions and potentiate the inflammatory reactions. So it's composed of complement 1, 2 through 9, and also there is a B and D. Those are the most common or the most known components. A cascade of activation will result after the antibody-antigen reaction, leading to production of several products of this system. These products can either help in opsonizing the bacteria, such as C3b. It helps to opsonize the bacteria. It means adding like a tag or a mark on the bacteria that this is a foreign subject that should be dealt with by the immune cells. So this product, for example, C3b, is actually functions as opsonization role. and we have several components such as C3A, C4A, and C5A. Those components, they can activate mast cells and basophils. Okay. Another component, which is the C5A, is actually can aggravate or initiate chemotaxis. Okay, attraction of white blood cells, most importantly the macrophages as well as neutrophils. Okay, another component, which is C5b, 6, 7, 8, 9, the long one, has a vital role in inducing lysis of cells. Okay, infected cells or the pathogen cells. T-cell activation goes through several steps and different characteristics that are different or is actually unlike the B lymphocytes. T-cell activation requires antigen recognition or presentation by another type of cells. However, B-cell lymphocytes, they don't require, they can get activated directly by the antigen, by binding to the antigen. So we need cells that are called antigen-presenting cells, such as the macrophages, the dendritic cells, which are specialized cells in presenting the antigens. They phagocytize, digest the pathogen or the microorganism and present the antigens on the surface, so that the T cell can recognize and bind the antigen, as well as the B lymphocytes. Those can work as antigen-presenting cells. T cells are actually classified into different types. We have the T helper type, which is the major in number, of course, and in function. It is the major T cell, T helper cells. Okay, and we also have T cytotoxic and T regulatory or suppressor. Those are known by markers, which are the type of the receptors. T helper are known by the marker CD4. Okay, cytotoxic is known by the marker CD8. So first of all, when the antigen is presented by antigen-presenting cells, it also requires binding to a protein that we will talk about later, which is called major histocompatibility protein. Also should help in presenting the antigen, okay, on the surface of the antigen-presenting cells. And once the antigen is presented, it will bind to certain receptors that bind specifically to the antigen on T cells, and that binding will activate the T cells. First of all, expansion and proliferation of activated T cell will result from this activation, and beginning with the T helper cells. Okay, so expansion of T helper cells, which play an important role in the whole immune response, not only the T cells function. Okay. And T helper cells will produce cytokines or lymphokines that can affect the function of various types of immune cells. And this will lead to expansion and proliferation of the cytotoxic as well as the T helper as a result of the lymphokines released by T helper cells. And both types also will save a copy that is going to be called memory T cells. Okay, so memory T cells will function similar to the memory B cells. They are going to be distributed in the lymphoid tissues all around our body, so that they can make the second response greater. The second response or the third response greater, faster, and way better than the first response. Okay. So this binding, actually, antigen receptor or antigen presenting cell, when digests the antigen, a molecule called major histocompatibility molecule will attach to the antigen and transfer it to the plasma membrane, where it can be identified and recognized by specific receptors on the T cells. Okay, so this is the way T cells get activated. Also, this actually takes place with the help of cell-to-cell adhesion molecules, which enhance the proximity of these cells to have the activation processed. Okay. So, as we know, B cells, they don't require any cell to be able to recognize their antigen. However, T cells, they need this type of molecules as well as cells in order to present the antigens and get recognized. And those are the antigen presenting cells, macrophages, B lymphocytes, and dendritic cells. So the major histocompatibility proteins are mainly two classes. The first class can present the antigen to cytotoxic T cells. However, the second class can present the antigen into the T helper or CD4 T cells. Regarding the receptors of the T cells, we might have on the same cell up to 100,000 receptors that can bind to presented antigens by MHC and antigen presenting cells. So there are many binding sites on the T cell to get activated. Okay? As we said, the majority of T cells are T helper, 75% of all T cells. T cells regulate the functions of other immune cells, such as the T cytotoxic, the T helpers themselves, as well as the B lymphocytes, and the other innate cells, such as the macrophages or the monocytes, okay, or the granulocytes, as an innate part of the immunity. This actually can be done by releasing lymphokines called or named by interleukin 2, 3, 4, 5, 6, and granulomonocytes, colony stimulating factor, as well as another cytokine, interferon gamma. Okay, all of these can affect the signaling and function of immune cells. There are subsets of T helper cells, subgroups of or kinds of T helper cells, named by Th1. Th1 or T helper 1 are actually involved in the macrophage stimulation as well as the production of the antibodies for lymphocytes, IgG. Okay? However, Th2 is involved in the allergic reactions, the basophils, mast cells, eosinophils activation, as well as stimulation of IgE, which are all involved in allergy. Okay? The last one is Th17, which actually involves the innate cells, neutrophils, and macrophages. You don't have to memorize here the cytokines that are stimulating or stimulated by these kind of cells, but you need to memorize these interleukins, each one, what is the target or what is the main function. So, for example, as you can see here from this schematic, this is a helper T cell, okay? It has been activated by binding to the antigen that was done with help of antigen presenting cells and MHC molecules, okay, as we know. This activation led to synthesis of lymphokines by the T helper cells. These lymphokines can activate the same helper cells, which makes a positive feedback on T helper cells, such as leukotriene 2. Okay? Also, leukotriene 2 released can also activate cytotoxic T cells as well as T suppressor cells. Okay? So it activates and accelerates their function. And furthermore, interleukins 4, 5, and 6, which are called B cell lymphocytes growth factors, okay, هذول المجموعة, released by the T helper cells. Those can activate the activated B lymphocytes, so it aggravates and potentiates their response. As we know, the immunoglobulins that are going to be released from the activated B lymphocytes, without the help or the lymphokines that are released from T helper cells, is going to be very low. Okay? However, with the effect or impact of the lymphokines, interleukins 4, 5, 6, by the T helper cells, the immunoglobulin release and the activation, proliferation, and number of activated B lymphocytes is going to increase to a great extent. Okay? Now, also another effect is their positive effect or stimulatory effect of the macrophages. Okay? Activation of the accumulation of macrophages. These interleukins, they slow down the motion or the activity of macrophages and rendering them, let's say, accumulated in the same inflammatory site. خليهم to accumulate, not to go away from the inflammatory site. They remain in the inflammatory sites, okay? And it increases their activation and, of course, their killing efficiency. Okay? So this is actually the activity or the function of the T cytotoxic, the CD8, okay, T cells. As you can see here, we have here 3 T cytotoxic cells. They bind specifically by antigen receptor binding, okay, to the pathogen that has antigens these cells are activated against, okay. They can release small protein or molecules that are called perforins. They poke holes inside the plasma membrane of the infected cell or the pathogen, and this will lead to lysis and removal or a flow of outflow of fluid, okay, leading to the destruction of the cell. At the same time, they can introduce chemicals, toxic chemicals, inside the attacked cells or infected cells or the pathogen cell in order to destroy them, okay? They destroy them and then they dislodge and go to destroy another cell. So they walk away and then destroy another cell several times. Okay. The cytotoxic T cells effect is actually done importantly for the infected, virally infected cells, okay, or to the transplanted organs, grafts, okay, transplantation from different tissues, from خلينا نحكي someone else or from another species, as well as the tumor or cancer cells. So this is important to get rid of cancer cells before they can, spread and metastasize. Okay, so one of the most important or one of the important parts of immunity is the regulatory unit or regulatory cells of T cells, which are called T suppressor cells. Those cells, T suppressor cells, are important for regulating the immune response, not to exceed or to be upregulated and be over-controlled or uncontrolled. Okay? زي ما حكينا, immune response, if it was severe and robust, it can cause deleterious effects on our own tissues. يعني ممكن يدمر التشوز أصلاً أثناء عملية immune response. Okay? So the suppressor cells, they suppress the actions of either cytotoxic or the other immune immune cells so that the immune response will not be upregulated to a level that is regulated or overreacting. Okay? And this is part of the immunological tolerance that we have. The term immunological tolerance means that our immune system has the capability to protect our own tissues, okay? And this actually starts from the pre-processing stage, or the mechanisms that we talked about, where clones of T cells are going to be mixed with antigens from our self-antigens, and then the clones that can be reactive against our own cells, our own antigens, are going to be destroyed. So this is part of how can we get tolerance, okay? Tolerance, immunological tolerance, so that our immune system will not fight our own tissues, okay? This actually can sometimes be disrupted or can go abnormally during normally مش normally هو pathologically during adulthood, okay? More commonly during adulthood. Some infections might cause generation of immunoglobulins that can identify epitopes on the same tissues, especially when the tissues are damaged and antigens of the tissues are released in large quantities. Okay, في حالة الانفلامation لما يصير في damage في الtissues, فالantigens, self-antigens are going to be released. Sometimes acquired immunity will develop against these antigens, self-antigens, and sometimes similarities or cross-reactivity between generated immunoglobulins, okay, and self-antigens may occur. هذا cross-reactivity may cause autoimmune diseases. بسبب الأوتوإميون ديزيزز اللي هو طبعا أمثلة عليها rheumatic fever, post-streptococcal, glomerulonephritis, myasthenia gravis, and systemic lupus erythematosus. A lot of other autoimmune disease can result either after inflammation, viral infection, or sometimes due to cross-reactivity between certain epitopes between the antigen and the foreign antigen or the bacteria antigen and the immunoglobulin that was generated. Okay, so also we talked about the immunization and the how can we make use of the properties of our immune response when we know that the second or the tertiary response is higher, then applying that by giving dead or killed or weakened viruses or antigens, then we can get a secondary response when we have a real infection by the pathogen. And this is actually the way or why we do immunization. Sometimes we use the passive immunity by giving the patient already made immunoglobulins. So their system or their immune system did not generate these immunoglobulins. Okay, passive immunity, when you give already made immunoglobulins from another, from experiment, from research, from another human being. So that's why it's called immune or passive immunity. Okay. And sometimes you get activated T cells, and you can give that to a patient where there is no immunization or where there is pandemic or some sort of diseases that are over-controlled. Okay, allergy and hypersensitivity also are one of the clinical perspective of the immune system when it's highly reactive, when it reacts to a greater extent than what it should. Usually these patients will actually develop immunity against particles or against things that we usually don't have immunity against, such as pollens, such as dust, okay, and or some chemicals This will lead to reaction and activation of basophils, mast cells, IgE, immunoglobulins. So this will lead to some complications, eventually leading to diseases such as urticaria in the skin or asthma in the respiratory system. اوكي اذا لهون بنكون احنا خلصنا اللي هي لاي اخر سلايد الالرجي. اوكي خلصنا السلايد. Today we will start with a new topic which is the blood types. Okay, blood types and transfusion. اوكي في عندكم اي سؤال على ال previous slides؟ اوكي. So, blood actually is composed of many components. One of the components are RBCs, the other we already we learned a lot about. In the old days or long time ago, when the science was not as much as يعني خلينا نحكي revealed as now, people or let's say physicians used to transfuse blood between patients, okay, without the knowledge of the antigens and the immune system that we already now know. So sometimes they notice that there are severe reactions and that can may be lethal to the patient you give blood. And sometimes it works fine and you save the life of the patient. Okay, so after knowing the science behind that, they discovered that our cells, all our cells, including our RBCs, they have several antigens that can elicit immune response, okay, if they were foreign to the patient or to the human that you are giving these cells or these RBCs, okay, and this immune reaction which we call transfusion reaction can be lethal, can cause death. And that's why we should learn the basic principle of transfusion and the compatibility between blood groups so that we can prevent these reactions and complications from happening. So they discovered that more than 30 common antigens are present, and some of them are rare. Most of them, they don't cause severe reactions if there is mismatch, but the main ones that can cause severe reactions if there were mismatch between the donor type and the recipient type were the ABO system, okay, the antigens that are related to the ABO system. We have two main antigens, antigen A and antigen B. And there are three genes responsible for the inheritance of these antigens. These antigens are present not only on our RBCs, on all our cells, okay? But because we are talking about blood transfusion, we are focusing about the RBCs. So the RBC, or let's go back, the inheritance of these antigens is related actually to three main genes, okay? The O gene, the A gene, and the B gene. A and B gene, each one will lead to the generation of an antigen, okay? To A and B respectively. So gene A will lead to the production of antigen A and B will lead to the production of antigen B. However, the gene O is like non-functional gene, so it doesn't make either A or B antigens, okay? So you either get an allele from your mom or your dad, so you have two alleles for each offspring. We have two alleles, one we get from the mother and one from the dad. So these two alleles makes the genotype, your genotype. Your genotype can be one of 6 probabilities, okay? حسب المother and father genotypes, okay, or mom and dad. The genotypes can be either 2 O alleles, and that will give us an O blood type, or you can have O and A, O from one of the parents and A from the other. So then you will have the A type because O is non-functional. So A is going to be the dominant, okay? So your blood type is going to be A. OB, your blood type is going to be B. And AA is going to be A, BB, A, and AB. This genotype will give you A and B antigens, so you are going to have AB blood type, okay? Now the story here is that each one of us, once you are born, okay, you will develop immunity against the antigens that you don't have, okay? And that occurs automatically without being exposed to the other blood group, okay? So even if you are not exposed, you didn't undergo any transfusion, your blood did not mix with any other blood, okay? After birth, you will develop immunity in your starting from your first month after birth, okay? And you will develop immunity against the antigen that you don't have. So if you were B, you will develop antibodies against A. If you were O, you will develop immunoglobulins against A and B, because those you don't have. صح? If you have AB, would you develop any immunoglobulins? No, because you have both antigens. So you will not develop any immunoglobulins against these per se, these antigens. So this immunity will develop. So if you have transfusion from the blood that it's not your blood type or compatible with your blood type, if you were transfused or you underwent transfusion from another human who has, let's say you are O and you got blood from A, then you are O and you have immunoglobulins against A. So transferring blood that have the antigen that you have immunoglobulins against will make immune reaction, okay? It will elicit immune response, robust immune response that will lead to transfusion reaction. And this is actually will complicate your condition. So normally, when we transfuse blood, you should transfuse blood from a type, okay, that you don't have antibodies against, okay? So O, you can give O to A or B or AB، صح؟ Yes، لأنهم ما عندهم إيش immunoglobulins against O، تمام؟ هلا في another system which is the rhesus or the RH system we will talk about later. Also we take into consideration not only the ABO system. Okay, now regarding the prevalence of the different blood groups, we have mainly the O blood group, which accounts for 47% from the population, and then the A blood group, 41% of the population. And the least goes to the B and AB, B group and AB group. Okay, this is actually shows the generation or production of the immunoglobulin with age against, زي ما حكينا, against the antigen that you don't have. هلا الانتيجين we call it agglutinogen because the antigen antibody reaction will lead to agglutination in the blood sample or the blood. That's why we call the antigen agglutinogen, and the antibody or the immunoglobulin we call it agglutinin. Okay? So the reaction, the transfusion reaction will occur because of the binding between the agglutinogen and its specific agglutinin. Okay? فلما نحكي agglutinogen, please just remember that it's the same as the antigen. Okay, so after birth, you will see that the titer of anti-A as well as anti-B. طبعا anti-A حيصير في الناس اللي هم group B or O. anti-B agglutinins راح يصيروا في الناس اللي هم A or O. So what is the reason why we develop immunoglobulins without even being exposed to transfusion, to the false blood group? Yes.
ممكن لانه بيجي عليه دم المذر لما يكون.
لا خلينا نحكي انه المذر اوكي هلا يعني ما في incompatibility يعني بين المذر وبين الطفل، ومع ذلك بصير في exposure مش exposure سوري بصير في immunoglobulin production. Yes.
ممكن نعتبر فرضا واحد هو جروب او البلاد تبعه وصار عنده انتي اي زي كانه صار عنده انتي بودي لاي شيء ثاني يعني.
لا هم سبيسيفيك لانتي اي. طب كيف عرفوا انه في شيء اسمه اي وعملوا انتي بوديز له؟
هلا انا قصدي انه البي سيلز نفسهم هم they produce later on plasma cells وبعدين في انتي بوديز، بتطلع انتي بوديز لاكثر من شيء، وممكن one of the things اللي بتطلع لنا
لا حتى لو احنا عندنا yes B cells clones that can identify A for يعني by chance اوكي عشان في كثير عندنا probabilities، بس ما حيكون في immunoglobulin titer عالي. هذا التايتر بصير اذا كان في صار في exposure already صار في illicit immune response بعديها صار في هدول الاميونوجلوبولينز بهذا الليفل اوكي مش من الفيرست response. لكن actually it was a mystery and there are several theories about that. One of the theories is that once the baby is born هلا before being born ما بيكونوا الانتيجينز developed. الانتيجينز ما بيكون developed. هلا اول ما يبلش الانتيجينز تصير على معروفه شو هي على الار بي سيز بصير في introduction للانتيجينز الفورين من either bacteria or from the food. يعني الفود ممكن يكون فيه antigen A antigen B ف then they will develop immunity against the one that they don't have لأنهم عندهم already tolerance من الإشي اللي عندهم إياه during the processing of T-cells صح؟ ف they only develop the immunity against the antigens that they were presented to them by either food or bacteria. هدول ال theories that are now being يعني خلينا نحكي more accepted. Okay? So at the age of 10 the maximum or the peak titer of immunoglobulins will actually occur and this will decline with age يعني مع العمر التايتر بيخف. طيب إذا زي ما حكينا غالبا ال environment exposure from the environment هاي most well accepted theory. The antibodies are mostly IgM بعدين IgG. هاي بعدين رح نستفيد منها في معلومة later on. Okay, إذا genotype OO is O blood type. They don't have agglutinogens, but they have anti-A and anti-B immunoglobulins or agglutinins. Okay, AB they don't have agglutinins, but they have the agglutinogens. Both the agglutinogen. Similarly, A will be they have anti-B and B will have anti-A. Okay, now how can we do the blood typing? Classically, they used to have a bottle of serum of anti-A and a bottle of serum of anti-B, and they mix the blood sample with each of those serum. Okay, and whenever you see agglutination reaction, that means you have the agglutinogen. Okay, so if you mixed blood with anti-A and you got agglutination, then this blood has agglutinogen A. If you mix it with anti-B and it did not do any agglutination reaction, then this doesn't have antigen B, so this is blood type A. For blood type O, you mix both sera with the sample and you didn't get any agglutination, so you don't have the agglutinogen. That's that's all. If you have the agglutination in both anti-A and anti-B sera, that means you have both agglutinogen, and that's AB blood type. You will see this in next week في physiology lab إن شاء الله. Okay, so this is the agglutination reaction that causes transfusion reaction, where red cells agglutinate, they plug small vessels, sacral blood vessels. It causes distortion and phagocytic attack. طبعا the distortion with red blood cells will lead to phagocytic attack and hemolysis. فبصير في تكسر في الدم. Hemoglobin will get out of the cells. Sometimes very immediate complement dependent hemolysis بصير. اوكي اذا كانوا التايتر تاع اللي هم الاي جي امز بالذات عالي جدا عن هذا الشخص، فبصير very fast hemolysis للار بي سيز بسبب الكومبلمنت سيستم. اوكي. Now let's talk about the other system, which is the rhesus system, the RH antigen or Rho D antigen. RH factor or Rho D factor, there are actually several common antigens, not only one antigens related to the RH system. They are named capital C, capital D, capital E, small c, small d, small e. Okay? هلا each one of us will have only one C, one D, one E, either capital or small. هلا anyone has the capital D will have positive RH. فعندنا هاي probabilities. من هدول مين اللي عنده ال RH positive؟ this one, okay, this one, and this one. هدول عندهم RH positive. So having the capital D. طبعا هذا مش معناه انه the other ones they cannot elicit immune reaction. They can elicit immune reaction if you have antibodies against, okay, but maybe minor or unnoticeable. The most خلينا نحكي serious reaction can develop from having the capital D, which we call D factor or Rho D antigen. Okay, so this is RH positive. 95 to sometimes 97% of population are RH positive. So very few, خلينا نحكي little percentage is for the RH negative people. RH positive people will not worry about having blood from either positive or negative. However, the negative ones, okay, here we should be actually, we'll keep attention not to transfuse RH positive blood to them. Even if you transfuse by mistake blood to an RH negative person for the first time, it will not cause any immediate reaction. Okay? It will not be life-threatening or any serious reaction. Sometimes you need exposures for the second or third time to get a severe reaction. Okay? Immunity is not like the case in ABO system. In ABO system, you already have immunity and immunoglobulins, and you will get transfusion reaction from the first exposure. صح? However, for the RH or rhesus, the RH negative will not have significant immune reactions. It will be delayed later. It will take weeks or sometimes months to have immunoglobulins against the RH positive, and then everything will change actually. اللي أخذته حيروح وبالتالي it will not cause any harmful effects. But maybe the second time you get exposed to RH positive, you will get a more severe reaction. Some people, a third exposure is needed to have severe reaction, transfusion reaction. نعم.
دكتورة هي نفسها اللي بتكون محطوطة مثلا واحد البلود تايب
minus أو positive.
يعني هاي هي نفسها.
Yes هاي هي. فبحطوا A مثلا positive أو A negative O positive أو A negative. It's negative sign هاي أو positive sign هي الريسس. اوكي. إذا زي ما حكينا بالفيرست رياكشن في delayed mild transfusion reactions, but you will get sensitized. There is a good probability you will be sensitized for further RH positive transfusion, and the agglutinin titer will increase only for يعني after months من الexposure. Okay, 50% 50% of RH negative are going to be sensitized by first exposure. يعني بعد first exposure بصير عندهم sensitization. يعني إذا second exposure حيصير عندهم transfusion reaction. 20% after second exposure. يعني بعد second exposure بصير عندهم sensitivity أو allergic reaction. Okay. Some people will not develop sensitivity. يعني ما حيصير عندهم immune reaction حتى بعد second response. Okay. هلا we care much about the RH, especially for pregnant women who are RH negative blood type and their husband is يعني خلينا نحكي positive RH. The child might have RH positive then. Okay? So if the child, the fetus, okay, during pregnancy is RH positive, and this is the first pregnancy with an with a يعني خلينا نحكي rhesus positive baby, you will, or this woman will not have any problem during pregnancy, okay? Because you know that the mom has never been exposed to RH positive and she does not have immunoglobulins in her blood against RH positive. So the first pregnancy will go just fine until the baby is born. Then the blood of the baby will mix with the blood with the mom. So RH positive RBCs, once the placenta is removed, removed from the mom, the blood of baby and the blood of the mom will mix. Then the mom will be introduced to the RH positive. She will develop immunity within weeks to months, okay? And then maybe in the next pregnancy, when she's pregnant with an RH positive fetus or baby, then the immunoglobulins in her circulation can pass through the placenta and attack the RBCs that are RH positive of the baby. They will attack the RBCs and that will lead to hemolysis, anemia, hemoglobin that will accumulate in the baby's tissues, especially the nervous tissue, and cause severe complications, which is a disease called erythroblastosis fetalis or hemolytic disease of the newborn. الاسم الثاني له. This actually occurs if there is no medical intention or intervention occurred in the first pregnancy. هلا how to prevent that؟ طبعا رح يصير في mental retardation ممكن يموت البيبي اصلا، اوكي؟ والعمليه بتستمر حتى بعد الولاده انه رح يضل الاميونوجلوبولينز لفتره معينه من حياته to attack their RBC، بيكون عندهم severe anemia. والهيموجلوبين بيخرب النيرفس تيشو تاعهم. جوينتس بصير عندهم.
حتى هل ممكن دكتورة كمان antibodies to pass through ايش اسمه؟
ABO system؟
لا during breastfeeding للولد لو عاش نفسه.
اكيد. هلا يعني هاي هي يعني خلينا نحكي ممكن to overcome this. But the already immunoglobulins in his blood is something يعني very bad. هلا how to prevent that? How to prevent that? Usually the mom is checked, and if there is a probability that she's RH negative and the child is RH positive, she will be given anti-ROD immunoglobulins against the antigen D just before delivery and after delivery, okay? So that these immunoglobulins will cover the RBCs of the child and she will not develop immunity against these RBCs. يعني بيعملوا neutralization لل RBCs بحيث إنه she will not develop immunity. And every time she gets pregnant with a RH positive child, they will do the same thing so that they will avoid the mom to make any immunity against RH positive. نمنع. Okay? So this is how to prevent the situation. How to treat the situation if already happened, فرضاً في مكان ما في أي نوع من medical intervention, then they can remove or replace by transfusion the baby's blood during pregnancy. خلال عملية البريجنسي، اوكي، اللي هو in uterine, or and also months after the delivery of the baby until the immunoglobulins from the mother are vanished لأنهم لهم lifetime بعدين بيروحوا بعد الولادة. This can minimize the complications that can occur, which is actually a very difficult procedure to do in utero transfusion for the baby. So this is the most important thing that I needed to discuss with you, and this slide is going to be the last for the second, خلينا نحكي the last lecture. Yes.
أنا مش إريثروبلاستوز ثالث هاي الألف ثلاثين وجه.
لا.
هاي ثالث.
هاي اسمها إريثروبلاستوسيس فيتاليس. السبب تاعها مش مشكلة بالهيموغلوبين. أوكي؟ الاسم القديم تبعها بس هي well known as Hemolytic disease of the newborn. السبب تاعها إنه في عنا immunity against RH positive RBCs من الأم. أوكي؟ هاي هي سببها. فبتسبب هيموليسيس بسبب الأجلوتينيشن رياكشن.
تمام؟ بس سؤال لكم هلا why we don't have similar disease for the ABO system mismatch between the mom and the fetus. يعني هلا ال ABO system ممكن الأم بتكون AB بتكون مش AB والبيبي ايش؟ اوكي ليش ما بتحدث هاي العملية؟ ليه ما يصير في خلينا نحكي إذا الأم الأم O والبيبي AB أنا هيك حكيت ولا لا؟ لا الأم لازم تكون O فعندها هي immunoglobulins against AB صح؟ والبيبي مثلا A أو B أو AB فرضا. اوكي؟ ليش ما تصير هاي البروبلم عند البيبي وسيفير رياكشن؟ لأن الاميونوجلوبولينز راحت على البيبي. سؤال تتذكروا الاميونوجلوبولينز لهم اشكال صح؟ الاي بي او سيستم الاميونوجلوبولينز المين اميونوجلوبولينز هم من البيج تايب الاي جي ام تايب. اوكي؟ فالاي جي ام تايب هو البرايمري خلينا نحكي immunoglobulin that is raised against الاي بي او او الاي بي agglutinogens فبالتالي they cannot cross the placenta. Okay, they cannot cross the placenta. Plus نفس الاجلوتينوجينز نفسهم بالبيبي they don't develop يعني على الار بي سيز الا في اخر ايام او بعد الولاده. بعد الولاده بصير عن جد في اجلوتينوجينز على their ار بي سيز. ففي تو يعني reasons for that. اوكي؟ طيب في عندكم اي سؤال؟ هدول المين ثينجز اللي احنا حبينا نغطيهم في هذا الموضوع. اوكي؟ بس يعني بدي أأكد إنه ال RH positive أوكي blood إذا حطيته ب RH negative recipient أول مرة إذا غلطنا وحطينا RH positive في RH negative recipient الدونر كان RH positive والrecipient كان RH negative بصير في mild and delayed transfusion reaction. ليه؟ لأنه بياخد وقت weeks أو months حتى يصير في immunoglobulins. ممكن بعدين هاي الاميونوجلوبولين تسبب شويه هيموليسيس لكنها بتكون ليتنت وبتكون مايلد. اوكي؟ فبعدين وي كان ميجر الاجلوتينز الانتيبوديز بعد اشهر ممكن نلاقيهم مرتفعين فنعرف انه في مس ماتش صار او في something immunity developed against الانتيجينز. لكن مش كل الناس they respond the same. 50% من ال RH negative are sensitized by the first exposure يعني خلص السكند exposure بيكون severe. 20% بعد السكند exposure حتى يصير في عندهم immune response. وفي ناس 30% non responders يعني still ممكن تكون انت من 30% اللي شو ما صار في exposure ما حيكون في immunity. اوكي؟